Okay, hold on, can you all see my screen? Yes, so, and I also like puns, so I have to make one right at the beginning. This is not your average talk for me because I'm actually not going to really talk about Notch at all, even though it's my most favorite cell surface protein. So I just kind of wanted to orient you into kind of what the lab works on, and then tell you about what the topic today. So yeah, I started in Notch. So the overall goal of our lab is really to try to understand the molecular details of how cells sense and respond to mechanical cues. And so we do traditional structural biology, single molecule force spectroscopy, kind of cell signaling to understand cell surface receptors like Notch and how they communicate mechanical force through their proteolysis. But then that also, as we started to study other cell surface receptors, we started to get into the field of molecular tension sensors because we really wanted to understand, what the forces these receptors were sensing in the context of cells. And then that's evolved into kind of tool development and developing assays where we can measure in high throughput the forces that cells are exerting. So because our overall goal now is to try to identify especially cell surface receptors that are driving certain normal differences in mechanical profile versus or diseased ones. And then we have this third kind of seemingly disparate area, which is using these proteins called HUH tags to link proteins and DNA together, which enable these other two areas of our research, but have also gotten us into lots of other kind of crazy applications. And I'm going to start here because I think these tags could be really useful to the mechanobiology community in general. And I think this forum will allow you to learn about these, but also we would love to provide recombinant proteins and things like that to anyone who might want to use them, especially in DNA tension probes. So protein DNA conjugation is kind of commonly used in all sorts of mechanobiology, and these are just a few examples here. But like in optical trapping experiments, if you're studying a protein of interest and you usually attach long DNA handles to it to isolate the protein from beads and surfaces and nonspecific interactions. People have used DNA nanoswitches to measure changes in ligand receptor interactions using centrifuge force microscopes. People have developed these really cool force actuators based on DNA origami, where you can apply tension to proteins attached to the DNA in very cool ways. Here's kind of another example of that that's kind of recent, where you can change the nature of DNA from single-stranded to double-stranded to apply a force across a protein. And then, of course, what we mostly use them for is attachment of ligands to DNA tension probes, which a lot of people here have been involved in. And so how do people normally attach proteins and DNA? So there's chemistry. A lot of times people will, you can get thiols or amines or click chemistry components on DNA and then use that to attach to peptides or proteins of interest. And an example of that is shown here. A lot of people in DNA tension sensor world use a cyclic RGD peptide as the ligand, which you can conjugate to DNA through chemistry. People in the Saleda lab, I'm showing a couple examples of how they use it. You can attach a biotin to your DNA tension probe and then have a biotinylated ligand like an MHC molecule or an antibody that uses streptavidin as a bridge to link those types of ligands to DNA probes. Shufeng Wang has many years ago developed protein G, which is a small bacterial protein that binds to constant regions of antibodies. And so that is shown here. Sorry, I can't quite see my own slide because of the, anyway. And so protein G, oops, did I just lose my presentation?
No, we can still see it.
Oh, I cannot see it on my screen. Hold on. Sorry.
No worries.
Okay, you can still see it. Okay. Yep. So yes, so the idea is that you have your DNA probe, you use chemistry to attach this protein G molecule to your probe, and then that allows you to attach any ligand that has an FC fusion or an antibody to this probe. So the problem, I mean, these are great methods, and some of the pros and cons of them are chemistry can be non-selective. Things like protein G are non-covalent. as well as biotin streptavidin. Sometimes you can't control the stoichiometry of the reaction. Oh, and people have also used like halo and snap fusion tags on their proteins of interest, and that allows you to form a covalent bond to the DNA as well, but you need specific modified DNA for that strategy. So we came across, when I first started my lab, this class of proteins called HUH endonucleases, which in nature, in viruses and bacteria, are involved with replicating single-stranded DNA. And so these these proteins in their catalytic mechanism of action. So first of all, they're called HUH because in the active site of the HUH endonuclease, there's a pair of histidines separated by a hydrophobic residue. And these histidines coordinate a metal ion that then polarizes a catalytic tyrosine to attack the phosphodiester bond of single-stranded DNA. So the first step is cleavage. Then this forms a covalent phosphotyrosine intermediate that obviously gets released in nature, but you can harness this for different biotechnology kinds of applications. And so you can basically use one of these HUH endonucleases as a fusion to your protein of interest. Then you just react it with a specific sequence of single-stranded DNA that's unmodified. Usually you need to append, 9 to 12 bases onto your sequence of interest to get this reaction to work. And then you get a covalent adduct between protein and DNA that's it's a very efficient reaction. The ones from viral replication-associated proteins are really efficient at forming these adducts. So the proteins are pretty small. You get a covalent link, although it is reversible under certain conditions. It's a very fast reaction, minutes. The conditions are very mild. All you need to make sure you have is the metal ion that the endonuclease needs. They're sequence specific. So the different families of these HUH endonucleases recognize different sequences of DNA. So this allows for orthogonality if you want to label two things at once. You can put them at the N or the C terminus of your protein. Importantly for mechanobiology studies, the catalytic tyrosine is pretty near, at least in the viral reps, near the C terminus, which allows you to design your system so that you're not too worried about the unfolding of the HUH domain if you're applying force to the rest of your system. And then the one caveat we've come across in our 14 years of using them is that it's been a little bit hit or miss to fuse them to proteins secreted from mammalian cells. I'll show you one example where it works pretty well when you fuse them to antibodies, but some other proteins we've had to do a lot more optimization. And we've done a lot of work in this area on all sorts of biochemistry and structure of these things, as well as different applications. So I just want to tell you very briefly kind of a couple of the things we've been working on in this area. So when we were working on these in an application for genome engineering, people were wondering, can you get these things to recognize a designer DNA sequence so that you could integrate into a certain spot in the genome, for example? But the problem was there weren't any structures at the time of these viral HUH endonucleases with their target DNA. So we solved those structures and found that the DNA kind of forms this U shape. And it forms this U shape around this specific motif in the protein, which we call the single-stranded DNA bridging motif. And this motif forms extensive interactions with kind of both sides of the DNA. And so we hypothesized that this was the element that was responsible for the sequence specificity of these proteins. And so my student Adam devised a yeast surface display method to see if we could alter the sequence specificity of one of these proteins. And in fact, we tried to engineer the protein so that it would covalently bind to RNA. And so basically, initially the wild type protein, if you look at the induction of the protein on the surface of yeast, and then on the y-axis we have a reaction with the fluorescent RNA, and so we want activity here to show that it's covalently binding to RNA. After a few rounds of engineering, we got a lot of signal in this upper quadrant, and then the resulting protein reacted very robustly with RNA. When the wild type didn't originally. And then you can see by the covalent adduct, the shift on SDS page, that our engineered mutant now covalently binds to RNA really well. And this is just some kinetics of a molecular beacon assay. We've also devised next generation sequencing based strategies to determine the sequence specificity profiles of a lot of these proteins. So we devised a method called HUH-seq, which you don't need to know the details of. And then we now have like a lot of these data sets. So Adam also wrote a program that anyone can use. If you want to use HUH tags in your research, it allows you to determine which sequences are best if you want to design an orthogonal HUH experiment. It will tell you like what sequence to append onto your DNA of interest. If you want to use HUH tags with a really long piece of DNA, it will search that DNA and let you know if you're going to have problems with nonspecific cleavage and things like that. Okay, so now we have mostly been using these HUH tags recently to attach ligands to DNA tension probes. So I'm just showing you DNA duplexes because that's mostly what we use, but this could be applied to all of the different cool hairpin and other DNA tension probes that people here have developed. So the idea is now you can take any protein-based ligand and fuse it to an HUH tag, and it can be a ligand on your tension probe, Notch ligands, even antibodies. You can also, due to the orthogonality, imagine using an HUH tag to attach to the kind of the anchor strand of the tension probe, which would allow you to immobilize these in more complex extracellular matrix environments. Like you could attach or fuse a collagen binding protein and immobilize them in 3D matrices, for example. So then where we kind of got into developing this high throughput assay is we were kind of new to the mechanobiology field when I was kind of starting my lab. And I feel like back then the questions in the field were kind of like, does this certain protein sense forces in the context of the cell and how much force? Then questions got a little more complex as time went on, and now people are asking things like, what are loading rates in cells, and what are the dynamics of force transduction, as well as, how does altering the microenvironment of cells or having a protein with a disease-associated mutation in it affect the force that cells are generating. And then also along the way, people were realizing that the mechanical phenotypes of cells are often different between normal and disease cells. And I think this is kind of a really cool graphic adapted from this company, Foresight Bio, where they use some sort of proprietary traction force microscopy to measure forces exerted by healthy cells and diseased counterparts. And you can see like disease cells can exert less force or more force. And so this kind of raised the question of, can you use a drug or identify the gene that's underlying or driving a malignant mechanical phenotype and then target that for therapeutics so that you could restore kind of normal tensional homeostasis in cells. And there are some precedents for this out there now. And so then the question for us kind of became like, which mechanosensing proteins are driving distinct mechanical phenotypes in normal cells or diseased cells? And so to do sort of drug screens or CRISPR knockout screens to identify new mechanosensing proteins, you need a high throughput assay to measure the mechanical phenotype of cells. And so we like to traction force microscopy is one of the gold standard ways of measuring the forces that cells exert on their surroundings. And it works by deforming a polymer embedded with fluorescent beads, and you can look at displacement of the beads to measure force. But molecular tension sensors, and I'm just showing one example, there are lots more examples using DNA hairpins and things like that. But DNA tension probes immobilized also can measure kind of traction forces of cells. And so this is just showing you the tension gauge tethers, which were developed by TJ Ha and Shufeng Wang. So these work by their DNA duplexes immobilized on a surface. They have a ligand that recognizes something on the cell. They have a fluorophore on them. And then when cells are plated on these probes, the DNA duplexes rupture with a certain propensity. And typically people measure this by looking at the surface that the cells are plated on and looking for, in this case, a loss of fluorescence. And you can kind of then see the forces that cells are exerting. So these were very attractive to us because they're pretty simple to use. And so we kind of started here and wanted to ask if we could develop a higher throughput version of this. I should point out that the Saleda lab has developed lots of other methods that were kind of floating around. One that kind of came out around the same time as the one I'm going to tell you about. But some of these are microparticle tension sensors where DNA tension probes are immobilized on microparticles, and then the microparticles light up when they interact with cells according to how much force those cells are generating. There's signal amplification of surface probes where cells plated on probes leave a scar of DNA, and then you can go in with reagents that will amplify the signal, and you can read out forces then by using a plate reader. And then this kind of cool method that's kind of similar to the one I'm going to tell you about, where basically cells plated on DNA tension probes release a cholesterol tagged oligo that has a fluorophore on it, and they actually tag the cell that exerted the force. And I'll tell you about our version of that. So again, we started with TGTs, tension gauge tethers, where these are immobilized on a surface. They have usually a ligand that binds to integrins on cells. And then when cells are plated on them, they will rupture a certain fraction of these duplexes. Typically, people read this out by imaging the surface, but we kind of flipped the question around and asked, could we actually look at the cell? Because this top oligo that gets ruptured has a fluorophore on it, and this is why we call these rupture and deliver rad-TGTs. This will get attached to the cell, and then due to receptor internalization, will we hypothesize, get internalized into the cell. And so the more probes that a cell ruptures, the more fluorescent it should be, and you could read this out by flow cytometry. And so again, I just kind of want to step through the process because these are pretty easy to use. Because the readout is flow cytometry, you don't have to be as careful about cleanliness of surfaces and things like that. So again, you plate cells on these DNA probes, and basically first we coat 96 well either glass or plastic plates with biotin BSA, incubate with Nutravidin, then incubate with biotinylated tension probes that also have your ligand of interest on them, wash, plate cells for 1 to 4 hours, and then dissociate the cells and analyze by flow. And so first did this work, and this is from our paper that was published a couple years ago. We first looked using traditional TGT imaging of the surface so that we would have something to compare our flow cytometry to. And so we typically use TGTs that have two different tension tolerances. So one is kind of the lower tension tolerance that, because of where the biotin immobilization is with respect to where the ligand is conjugated, the DNA ruptures by unzipping. And that occurs at a lower force than if you have the biotin on the other end of the anchor strand, where now the DNA ruptures more by shearing, and that occurs at higher forces. And there are all sorts of variations in between, but these are the two that I'll typically be telling you about. There's unzipping and shearing. Again, we're attaching ligands using the HUH tag, and the HUH tag we're using usually is called WDV. And so usually in most of the experiments I'll show you, WDV is always the negative control. And so in this first experiment, we made a WDV fusion with a fibronectin domain that presents an RGD loop, presumably similarly to like a cyclic RGD peptide. And then we used this fusion to a protein called echistatin, which is derived from a toxin, but it binds to all beta-1 integrins with sub-nanomolar affinity. And so you can see that now these are, I think, U251 cells, or maybe CHOK1, I can't remember. This was in the paper, plated on top of these TGTs. So you can see that with fibronectin as the ligand, you do get rupture of some of the unzipping TGTs. And it looks really bright here, but actually the cells don't spread very well, and they're really small. And a lot of other people have seen that as well. Whereas on the shearing higher force TGTs, you see more patterns of focal adhesions, which others have seen. the kind of nice thing is that this ligand equistatin, you see really nice cell spreading in both unzipping and shearing TGTs. And so then when we quantified that by flow, this is what we get. So this is a histogram where we're looking at Cy5 fluorescence and the cell distribution. And so WDV is our negative control, and you can see there is a little increase in signal on the fibronectin ligand for the unzipping, and then a lot bigger increase with the equistatin ligand. So this seemed to be working. And then what I'm going to tell you about the rest of the time is that we, even though we published the assay a couple years ago, we've now spent a lot of time trying to tweak the assay so that it would work for all of the different cell types we wanted to study and all of the different ligands that we wanted to use to probe cells. And so I kind of have all of these, there's a little bit of validation, a little bit of kind of interesting features that some of you might find interesting, and then maybe a little bit of utility. In the end, I'll get to a little bit of biology. Okay, so first we wanted to test if our signal was sensitive to disrupting the force driving machinery in cells. So we treated glioma U251 cells with blebbistatin, and you can see, again, these are the flow histograms with the negative, the positive, and then when treated with blebbistatin, you see a decrease in fluorescence or a decrease in cellular force, relative cellular force. We also had CRISPR knockouts in U251 cells of talin, which is a key component of the focal adhesion machinery, as well as a cell surface protein called CD44 that binds to hyaluronic acid and is important in cell migration and things in glioma cells. And so the parent U251 cells have this force profile. Knockout of CD44 decreases that force, and then talin more significantly decreases the force. And these are on unzipped probes. We also had vinculin knockout cells where, and then we tried to rescue the vinculin knockout with vinculin GFP. So here is the WDV negative control. Here is the force profile, mechanical profile of wild type MEFs. And then here are the vinculin knockout MEFs here, where the force, this is in the shear, on the shear probes, the force decreases almost down to baseline. And then transfecting in increasing concentrations of vinculin GFP kind of increases that force, although we never get back to the wild type mechanical profile. So it seems like our assay is detecting relative cellular forces. One other kind of interesting feature is that I've been telling you about reading out the fluorescence in the cell using flow cytometry. And so here is kind of a slightly different version of what I showed you before, where now the fluorescence is on the y-axis in fold change over the WDV control. So where fibronectin had a little increase in signal and the equistatin ligand had a larger increase in signal, if you attach a DNA barcode onto the top strand of the TGT, you can also quantify the ruptured oligos in the cell using sequencing. And so we kind of did the same experiment and read it out by sequencing as well. And so I think here is showing you Sanger sequencing, and here is next-gen sequencing. So we basically, after the experiment, PCR'd the DNA in the cells and then did sequencing, and you kind of get the same profile. And so this can be really useful for different kinds of multiplexing versions of the assay. Okay, one thing we ran into that I think everyone who works with tension sensors has probably run into at some point is the prevalence of nucleases. So nucleases can be expressed on the surface of cells. There are nucleases in serum. And so Xufeng Wang had developed this surface nuclease sensor and also started using peptide nucleic acids as his anchor strand. And so these surface nuclease sensors are basically TGTs that don't have a ligand on them, and they light up when the DNA gets cleaved by nucleases. So Matt, I forgot to mention, Matt Pollack did a lot of this rad TGT work. I think I showed him his picture at least on a previous slide. So he used this surface nuclease sensor in just kind of normal conditions in a cell line that doesn't express a lot of surface nucleases. And then in the same cell line using serum in the media, you see a great increase in kind of background fluorescence because there are nucleases there. And then in a cell line like C2, C12 myoblasts, they are known to express surface nucleases. You see a great increase in signal. And so these are some of the cell lines that we wanted to study. So we have adapted our assay and it does work. We typically use the peptide nucleic acid anchor strand now to mitigate these nuclease problems. As I told you before, the HUH tags allow you to use any ligand on the DNA tension probes. And so why might you want to use other ligands besides cyclic RGD or echostatin or something like that? I mean, beta-1 integrins are only a small subset of the integrins present on cells. And so if you want to study immune cells, for example, you need to use different ligands. We also think that if you could use different ligands on TGTs, you might be able to access kind of higher dimension mechanical phenotypes, which might help you distinguish cell populations by mechanical phenotype. And if you want to study other mechanosensing receptors besides integrins. And so we have kind of used HUH tags to attach other kinds of ligand besides standard proteins. So we have fused HUH tags directly to the heavy or light chain of antibodies, and then we recombinantly expressed them and purified them, and then attached them to TGTs. And this gel just shows you the HUH reaction with two different antibodies we made, one a K20 anti-integrin antibody and one pertuzumab, which is an anti-HER2 antibody. And you can see this shift up in the SDS page to show that in this context, the HUH reaction is working well. And I'll show you some data on that in a few slides. But we wanted to be expressing antibodies recombinantly can be kind of a pain. So we wanted to be able to just take any off-the-shelf antibody and attach it to TGTs. So we started with what Shufang Wang had done. I showed you many slides ago, where you can we could make a fusion of protein G with our HUH tag, and then that should allow us to attach any antibody we want to the TGT. We did run into some problems, though, with the non-covalent nature of the protein G antibody interaction. So Matt ended up making a protein G that has a photo crosslinker in it. And so that allows you to then shine light on your protein G antibody complex to covalently attach your antibody of interest. And just to give you a sense for that, he was looking now at cell spreading area under different contexts of TGT antibody adducts. So in the first lane here is the covalently attached antibody to the TGT. And you see a nice cell spreading in both unzipping and shearing conformation. But when you have, he had protein G alone without the photo cross-linking, the cells kind of balled up and didn't stick very well, suggesting that the protein G antibody had dissociated and the cells didn't have anything to stick to. But when you introduce the photo cross-linked antibody, you see a restoration of the cell spreading, especially in unzipping, not quite in shear, but better than without the photo cross-linking. So I'll show you a piece of data with that as well. So Matt, in collaboration with the Sitochi lab at University of Minnesota, we've been profiling natural killer cells because they are interested in asking how exhaustion, for example, affects cellular mechanotypes of these natural killer cells. They also want to profile different chimeric antigen receptor natural killer cells to see if the mechanical profiles of the cells have any correlation to clinical properties of these kinds of therapies, like invasivity or and things like that. So Matt took patient-derived natural killer cells from 2 donors. And so in navy blue is our kind of negative control. The light blue is showing you the protein G antibody. This is an LFA, anti-LFA antibody, which is the integrin on T cells and natural killer cells. Without photo cross-linking, you see a little increase in signal, but when you do the photo cross-linking, you see a nice distinction between the negative and the positive control. So now we're studying some of these exhaustion questions and things like that. Okay, I'm doing okay on time. Okay. The other thing is that these rad TGTs can be multiplexed to allow you to separate mixed cell populations by mechanical phenotype. So I'm going to show you two examples of that. So you can co-plate TGTs that have different tension tolerances. So here we have the shear labeled in green and the unzip labeled in with Psi5. And we have mechanical profiles of CHO-K1 and U251 cells. And then you can kind of mix them up and you can clearly distinguish cell populations. This is kind of an extreme example, but this could just be useful if you want to use this in the context of screens and things like that to be able to separate populations. And then we've also multiplexed by ligand. So here we have TGTs attached to an integrin antibody on one axis and a HER2 antibody on the other axis, and then profiled several different cancer cell lines. And you can see that you can kind of separate out these different populations of cells, which would have been more difficult just using a 1D kind of a readout. Okay, so finally, as I told you before, like these disease cells often have different mechanical phenotypes than normal cells. And so people are starting to develop mechanotherapeutics as drug targets. And so we were wondering if rad TGTs could be used as a readout of how drugs affect the mechanical phenotype of cells. And so the idea would be to titrate the drug and look for differences in the rad TGT signal. So, and so we just put this on bioRxiv a couple weeks ago. So integrins have an inactive conformation and an active conformation, and there are many modulators out there. Some have been FDA approved, some going through FDA approval to target integrins in different diseases. And so I'm just going to show you the ones for LFA1 in the context of natural killer cells, but we've also profiled glioma cells and some of the beta-1 integrin modulators. So the idea here is that you first take your cells and incubate them with different increasing concentrations of the modulator, and then you plate them on the rad TGTs, and you again read out the fluorescence of the cells and plot them. And so without drug, you have your normal mechanical profile of the cell. And then depending on whether you add an activator or an inhibitor, you would see a shift in this cell population and that you can plot. And so for the natural killer cells, we saw for an inactive state inducer called TS1, you see a decrease in signal, a concentration decrease in signal. And then for two different, well, an active state inducer and an active state stabilizer, you see kind of this increase in signal, and you can calculate IC50s and EC50s. And so we think this could be useful just generally in the development of mechanotherapeutics. Okay, so finally, onto a little bit of biology. So now that we can, we've used PNAs to mitigate all of the nuclease activity, and we can attach different ligands. We wanted to actually start asking some biological questions. And so with our collaborator, David Largaspada at University of Minnesota, we are studying this disease called malignant peripheral nerve sheath tumors. And so this disease in most patients starts with patients who have a deletion of NF1, the NF1 gene, which encodes neurofibromatosis. And so these patients develop on the nerves these kind of benign but kind of large tumors. And then other genetic alterations are known to happen that eventually can lead to this metastatic cancer. And so the kind of attractive thing about studying this disease and looking at links between mechanical phenotypes and genetics is that these genetic stages are pretty well defined, and there are decent cell line models. There are decent PDX cell lines available from patients. and different mouse models. And our collaborator, David Largaspada, has also stably introduced Cas9 into like all of these cell lines so we could do some of the CRISPR screening and things that we had been interested in. So we first decided to measure the mechanical profiles of the different cell lines representing the different genetic stages of the disease. And so we saw that first in parent Schwann cells have, a certain mechanical profile, a decent fold change signal above the WDV alone. And then the NF1 knockout cells have a much increased fluorescence or cellular force exerted compared to parent Schwann cells. Then in the progression from here to the malignant tumor lines, there seems to be a progressive decrease in mechanical profile or number of duplexes ruptured or force. And so we have a few hypotheses that we're trying to test related to these changes in mechanical force. So first, NF1 loss. So the hypothesis here, so NF1 is a tumor suppressor gene, and so it restrains RAS. So when NF1 is lost, you get unrestrained RAS signaling. And increases in RAS signaling have been tied to cytoskeletal changes through like the ROCK pathway. And so we hypothesize that this NF1 loss is due to unrestrained RAS signaling. A lot of the further genetic changes have to do with mutation of this polycomb repressor complex in cells. And so we have data showing that the loss of this polycomb repressive complex alters a lot of growth factor signaling and integrin expression. And so we hypothesize that that's why then the forces decrease through further genetic manipulation. So to start to, I'll show you kind of some data on this NF1 loss hypothesis. So again, back to kind of the canonical view of our assay readout. Here's our negative control. Parent Schwann cells have a certain mechanical profile, and then NF1 knockout, the cells have an increased fluorescence. So we hypothesize then, if this is due to unrestrained RAS signaling, that we should be able to phenocopy this result by introducing a different kind of mutation that would lead to unrestrained RAS signaling. And so now I'm back to showing you kind of fold change in Cy5 fluorescence. So again, NF1, you see knockout, you see an increase in fluorescence. And then if you introduce the NRAS G12V mutation, you see an even further increase in cellular forces. And then combining both of the mutations, you see an even further increase. So now we're using drugs downstream of some of these pathways to kind of further test the mechanism of this. And then same with the PRC. Again, we have some surfaceome cell surface profiling data of cells that either do or don't have the PRC2 complex intact. And we see drastic changes in integrin, CD44, other cell surface receptors. So we're now testing, targeting some of these receptors to see if they are the ones driving the mechanical phenotype towards lower forces in the progression of the disease. And then finally, we really think, and a lot of these high throughput assays, the really kind of cool thing about them is that now the mechanical force that the cell exerted, because the cell is now tagged by fluorescence, the mechanical history of the cell is encoded in the cell. So this can allow you to flow sort cells according to mechanical phenotype. And so you can start to explore mechanical heterogeneity in disease. So we have measured mechanical profiles of PDX cells from forms of glioblastoma. And you can see for these two PDX cell lines compared to a parental immortalized glioma cell line that the mechanical profiles are a lot broader. And so we are trying to ask if the cells out here that are exerting higher forces have different transcriptomic, epigenomic signatures compared to the cells that are exerting lower forces, and kind of trying to get a sense of if it's true mechanical heterogeneity. And then again, we would really like to use screening by mechanical profile in some CRISPR knockout or drug screens to identify key receptors. And so we've done kind of a pilot CRISPR knockout screen in Schwann cells, where we transduce cells with a very small random CRISPR library that we got from Addgene. And so just what I wanted to show you, because we still have a lot more to do on this, is that the mechanical profile of the cells that have been CRISPR screened have a much broader mechanical profile. And so we're really wondering if some of the cells with exerting lower or higher forces will identify some important new mechanosensing genes that we might be able to use to target for MPNST or other diseases. So in conclusion, so HUH tags allow basically any ligand to be presented on TGTs. Even if you want to express a mammalian protein in fusion with an HUH tag, we have some tricks. If the reaction doesn't work right away, we can provide some of the tricks we found to get around that. We would love to provide recombinant HUH fusion proteins to the center for other people to use, because I mean, the point of the center is to disseminate mechanobiology and make it easier for anyone to do it. So we would be happy to do that. So I told you about these rad TGTs, like some of the features here. And that's about all. And I'd like to thank my lab. Matt Pollack was really the pioneer of all of the rad TGT work. And Aaron, Matthew, and Andrew have also worked on some TGT studies that I didn't really show you their data, but I'm happy to take any questions.
Awesome, thank you. That was great. That was great. So we'll open it up for questions. If people want to leave questions on the chat, I can try to read those, or Wendy, if you can read those. And then if you want to ask a question, you can unmute. I see. Ah, of course,
Chang.
Chang. Chang already has his hand up. Okay, Chang. Go
Hi,
right
Chang.
ahead.
Hey, Wendy, great talk. Really enjoy it.
Thank you.
I actually have some technical questions. I'm going to ask much just the surface, and then maybe I'll send my research scientists to reach out. So I was hoping to see you to hear your talk on Notch, but this is great.
I can give one next year on Notch. We have been working a lot on Notch lately, so we've been
Yeah, so
yeah.
so so this is I guess the question touches both Notch and this HUH chemistry. So you mentioned that you are trying to make the method generally applicable to any ligand you want, any cell that you want. And I guess not being a chemist, I naively think that it works on one, it works on the other, and I found out not so. And so now we've been using synthetic notch as a platform, right?
Uh-huh.
You know
Oh
knows
yeah,
that very,
I
very
did the
well,
first in
right?
Notch experiment.
So we have one walk, right? An antibody, and we try to do others, it doesn't work. It express and we know that it binds to the counter molecule, but it does not activate the knots. So do you have any
Yeah, I have a lot of
insight?
thoughts. I have a lot of thoughts on that. I mean, I think Syn Notch is really nice because it's very modular, but I think in the Notch signaling field, it's becoming appreciated that the ligand binding domain, which is this long, it's made up of 1,000 or 36 EGF repeats. That it's playing some role in Notch activation as well. Notch seems to form these signaling synapses, kind of like in immune cells. And I think in SynNotch, depending on the affinities of your ligand, you might not be kind of recapitulating the canonical physiologic notch signaling.
So it's not just pulling on it and that's it. It
I mean, I
actually
don't think
require
so. I
other
think,
things.
yeah, I think there could be other things that are required. Yes.
Ah, yeah, that's what I wonder, but I hear from you that really validating the thought.
Yes,
So
I would
so
love to talk more about that at some point.
okay, I'll
Like
let
my favorite
others to
topic.
ask question. Thank you very much. It was great
Yep.
talk.
Great. Maybe I'll ask the next question. I have a whole sheet of questions here, and But one thing that I sort of was playing in my head is kind of this really interesting observation that when you link fibronectin to HUH on the TTT, you see low levels of tagging in contrast to these really high affinity ligands.
Yes.
And so the natural question in my head, of course, is why, right? And the only thing that comes to mind is the propensity that fibronectin integrin bonds have been shown to display catch behavior, meaning that affinity goes down. So do you think the fibronectin does get pulled off and but then because it's no longer under a mechanical load, it dissociates and falls off? Or there's some other aspect to what's happening that leads to this lower level of tagging?
Yeah, I mean, I think with fibronectin and kind of any normal affinity ligand, you probably have this like on and off rate of the ligand binding interaction as well. And then it's just going to depend on whether it's engaged when you're pooling. Whereas I think ligands like echostatin and maybe even antibodies, which are much higher affinity, they're more irreversible kinds of bonds. So it's like you engage and then and the cells on echostatin don't move around as much for sure as on fibronectin.
Yeah.
And I mean, I know one criticism of that is like, well, are you like really looking at something physiologic then? And I mean, I always answer that we're trying to it's a relative mechanical phenotype kind of assay, and we're looking at how, at how certain drugs or gene knockouts or whatever affect um, the mechanical phenotype. But yeah, certainly echostatin is like not your normal integrin ligand, so it's going to behave differently. And I love the question about like whether there's catch bond, slip bond behavior. The same thing I think that addresses Cheng's question as well. A lot of these SID-Notch kinds of ligands are antibody antigen interactions, which are going to have very different properties. Like the Notch delta interaction is like low micromolar for the molecules by themselves. So clearly, like there's something about the specific catch bonds that might be going on here that
So,
enable
so,
further signaling.
so Wendy, do you know that we have made a whole bunch of notch receptor catch bonds with Bob Harlinger?
Right. Well, I remember your paper with Chris Garcia and everything too, like in using the
That was
BFT.
old days, but we have a whole
Oh,
bunch
you
of
have new ones
unpublished
with the
days.
glycosylation mutation.
I thought he presented in the Notch meeting, but the postdoc making those measurements has gone, so it's like sitting somewhere in my
I'll
computer.
catch him at the next Notch meeting for sure.
All right, I have more questions, but I see Allison has her hand up, and then I know Bian Chao has a question in the chat. So let's do that. Allison, you go first.
Hi, I'm a trainee in Matt Lang's lab. I was
Oh,
just curious, once you put an HUH tag specifically on like a longer piece of DNA, what would be the means of purifying possible excess, whether it's the DNA or the HUH tag? And because that's usually like personally my biggest issue is being able to
so
purify things.
yeah, would
Yeah.
this be like a long double-stranded DNA handle and then maybe with the
Yeah.
single overhang?
Yeah.
Um, yeah, that's a good question. I mean, I guess my sense would be that you would want to have the HUH tag in a huge excess, and then you could
Mhm.
just use normal like DNA purification columns to,
Okay.
um, because it's a pretty efficient reaction. We haven't, I guess, systematically tested it for like some long
Would
handles
there
with
be a concern of like the ethanol that would be present in those DNA purification columns, like in the wash steps to
for the
denature?
protein.
Yeah.
Yeah, I mean, I think you probably would denature them slightly, but I don't think
Okay.
I'm sure that they would refold
Okay.
when put in normal buffer conditions.
Thank you.
Plus the conjugate would be made so you don't need the function as much anymore, like the folding
Right.
of the protein.
True. Thank you.
Great. So Bian Chao, I see you want to ask your question and unmute?
Sure. Wendy, really nice talk. So it's really interesting. You convert force readout from fluorescence imaging to flow cytometry, right? So one of the key assumption is that the oligos or the fluorescence fragment of the oligo, once it peel off from by the force, it get internalized, so it can be read out by the flow cytometry.
Yeah.
How sound is that assumption? Will some of the oligo just dissociate and the receptor not internalize?
Yes, I mean, we did, one of the reviewers, were you the reviewer? One of the reviewers asked us that on our paper.
I'm not the reviewer.
No, I mean, I think at least for the Sci-5 fluorophore, which I don't think is a pH sensitive one, I think there were some oligos still stuck on the surface of the cell, which contribute to the signal, but those were still oligos that were ruptured. We do have a quencher on the anchor strand, so that sometimes cells generate so much force that we do see the whole duplex ripped off of the surface, and we have a quencher on the anchor strand to try to mitigate that a little bit. But yeah, I think some of the oligos can be still stuck to the outside of the cell and not internalized, but we're kind of reading them out at that point, and so it's an additive effect of the signal. It was maybe like in the study we did after 3 hours, it was maybe like 10% of the signal was from oligos that were on the surface, and we used something to chew up the surface DNA that couldn't get into the cell to measure that. But that's a great.
The thing is, if it's stuck to the surface, you can still read out by flow cytometry. It still gives the signal. The question is, once it ripped off from the surface, whether some are internalized, will keep there, but other may dissociate during the process, whether those signals are not read out.
Yeah, I mean, I think that could, I guess, be an issue for some normal affinity ligands. I think for echostatin, it's so tightly bound that it probably doesn't associate, but that's a great point. We should do kind of the same studies for kind of some normal affinity ligands.
Thank you.
Brent?
Hey, Wendy, great talk.
Hi.
I have a big thought, potentially unfair question. So here we go. So I think I know you can handle it. So, I think when we think about mechanosensitive diseases, right? I think there's at least two options. Like is it a cell-driven, cell-autonomous disease? Like the cell is actually more contractile? Or if you talk to maybe some of the ECM people, it's all these are diseases of the microenvironment, right? So the cell's fine. It's just in the wrong neighborhood. Right. And so I guess this is what got me this is you showed you could use the HUHs to tag on either ends. Right.
Yeah.
So I'm assuming when you're doing this really, I love the rad TGT assay. We talked about it before. When you're doing this, is it kind of on TCP? And if it is, can you, start changing that surface to maybe come at this question two different ways?
Yeah, I mean, yes, I don't remember if I went to the ECM meeting, the Gordon conference this last summer, and got a lot of questions like this. And yes, my dream is to really like adapt these, because the nice thing about the flow readout is if you have a 3D ECM that you can extract the cells from, like the readout should be the same. And absolutely, and Dave Odie here has done all of the stiffness of the surroundings, really affect the contractile forces of the cell. So kind of the stiffness sensing, we should really be looking at for all of the cells, because we're just using glass. But yes.
Yeah, or you can skip a step and just go to the viscoelastic sensing, because that's what everyone's
right.
I'm teasing, sort of, but
sort of.
Anyway, great talk. Thank you very much.
Great. I'm going to ask another question related to it's a big picture question, right? So when, for example, you show these really nice results with the Schwann cells, and different treatments or different mutations, the NRAS mutation, for example, was modulating this force, and then you show this nice volcano plot. But then the question that emerged in my head is like, when you see a signal change, is that because the copy number of the receptor, which I think you have evidence the copy number of receptor change, but it could also be that the cytoskeleton, the motor driving these forces, is altered. You could also imagine that there's differential glycosylation in the integrin, so the affinity between the receptor and the ligand you're using could change. Right? How do you how do you disentangle between these potential mechanisms that drive the observation?
Yeah, well, and is it I mean, every time we study a new protein, we find that they're all connected up to integrins like CD44 and dystrophin, dystroglycan. They all inside of the cell are connected up to the same actin network. So is it ever really going to be one receptor that's driving a certain process? And yes, I mean, we hope to start to get at some of those questions by doing some of the knockout screens, how many candidates will we come up with once we analyze the results of a CRISPR screen, and do they all just go to one kind of node inside of the cell, and maybe that's what you want to target instead of an individual receptor. But yeah, great question.
Yeah. So, but I mean, I thought perhaps your screen suggested that it's a reflection of diminished integrin expression, surface expression, but
Yes.
you think it could be other mechanisms.
Yeah, I mean, and I think, I mean, there were lots of things that changed, some intracellular proteins as well. So yeah, I think and plus it's like a different transformed cell line, in that case. So, I think there could be lots of things that are going on.
Interesting. So, I mean, it's like when you do the vinculinol, the vinol maths, right? I mean, so there it's presumably the integrin copy number doesn't change, but it's that coupling, the transmission of the
Yeah.
force and the maturation of the focal adhesion that's driving it. But
Yeah,
yeah.
well, and in MPNST, there are tons of there's tons of dysfunction in DNA repair and in chromatin organization and everything. And so like we're hoping to get at it that way too, by sort of analyzing some of those cells that are exerting different forces.
Mm-hmm. That's neat. Um, oh, I see one more hand up. Maybe this will be the last question because I see it's 1 o'clock. Sasha,
Yeah.
do you want to unmute and ask your question?
Yes, first off, thank you so much for the talk. It was very nice. So is the shearing versus unzipping behavior dependent on where the nutrividin is?
Um,
How does it work?
Yeah, I mean, in DNA duplexes, it can be, you can alter the GC content, and that would also alter the kind of rupture force. But yeah, in this case, it's not it's where the biotin is located on the anchor strand. It can be on one end that's kind of like right under the ligand, and then it will unzip, or it can be completely on the opposite end, and then you can imagine the DNA shears, and then it can be in between. And I mean, I'm talking about it like it's a digital thing. I mean, probably both of these behaviors are going on and contributing to the tension tolerance, but yeah, so it has to do with where the biotin is anchored.
Thank you.
Thank you. Thanks, everyone.
Yeah. All right. Well, yeah, thank you, Wendy, again, for great seminar and great discussion afterwards. I'm happy to hang out here
Sure,
actually,
we
do
can
you mind
chat.
hanging out for just a little
Yeah,
bit longer and chatting?
sure.
Well, and
Yeah, sounds good.
thank you, everybody else.